Today's talk title is Endogenous Retrovirus and Type 1 Diabetes, Conserved and Mutated Epitopes of the Virus-like Gag Antigen, and it will be presented by Yang Dai, PhD, coming to us from the HERV Lab in San Diego. A little bit about him. He has extensive experience investigating autoantigens and isolating autoantibodies and T cells in organ-specific autoimmune diseases. His work centers on experimental autoimmune thyroiditis, EAT, and type 1 diabetes, examining pathways that modify autoantigens or alter antigen processing and presentation of T cell epitopes, particularly thyroglobulin and GAD65. A major focus of his research is the role of exosomes in type 1 diabetes, and he's published 3 key studies showing 1, the pro-inflammatory function of T cell antigenicity of exosomes, 2, T-independent B cell responses to exosomes, and 3, activation of memory autoreactive B and T cells in NOD mice by exosomes from islet mesenchymal stem cells. These findings position exosomes as a novel antigen source in autoimmunity, and more recently, his group identified endogenous retrovirus antigens, otherwise known as ERVs, GAG and ENV, highly enriched in islet-derived exosomes. Only NOD mice, not T1D resistant strains, expressed ERV antigens in islet cells and their exosomes. GAG stimulates autoreactive T cells confirmed by shRNA knockdown, and these results suggest ERV-containing pseudovirus or virus-like particles drive autoimmune responses. Because ERV antigens exhibit multiple genomic copies and sequence variants, his team developed targeted deep sequences to identify these variants and quantify open reading frames. And using this method, they've mapped dominant T cell epitopes and tested whether GAG variants encode altered peptide ligands presented by MHC molecules that trigger autoreactive T cells in NOD mice. Thank you so much, Yang, for joining us, and we really look forward to this talk as you sort of unpack your newest work.
Thank you, Monica. Have this opportunity to share our studies, and thank you all for being online to listen to the talk. I work in a small nonprofit unit in San Diego called the HERV Laboratory, and we share the resources and the facility with a biotech CRO company called Biocedia. As you may agree, Treg cells are particularly important for T cell mediated autoimmune disease, and including type 1 diabetes. I'm thrilled to see that this field is awarded with Nobel Prize this year. And before Sakaguchi identified CD25 surface markers and subsequently FOXP3 intracellular transcription factors for this population of T cells, immunologists already know the T cell subsets can have suppressive functions for other immune cells, including T effector cells. And those time we called it T suppressor cells. This including Richard Gershon, Iran Cohn. They describe the T cell regulatory network. And of course, my mentor, Eli Sukas. And Eli's focus is the peptide determinant that recognized by the regulatory cells, or are they different from those recognized by the effector T cells? So, and those early pioneers, they are different from modern immunologists. The focus for early ones, they study simply outside of T cells or partners of the T cells, and the modern immunologists, they study what's happening inside the cell. So that's an easy way to see the difference. But studying the inside, of course, outside is called antigens, especially the antigens are very challenging because T cells are known for polyspecific and degeneracy and the large repertoire. So cross-reactivity is a challenge for studying the T cells. For me, when I study in my lab, I've always focused on what's the antigens, how T cell reactive to the antigens. So when I was in Eli's lab, we studied GAD65. The C-terminals of this autoantigen is very special. They are enriched with antibody epitopes, CD4 T cell epitopes, CD8 T cell epitopes. In this C-terminal region, they are highly susceptible for protease digestion. So maybe the structure nature make this region become a dominant target for T cell and antibody response. We produced T cell hybridoma clones for this region because we focus on class 2 MHC due to their tightly association with the disease. So, and the different clones can recognize the same region from 524 to 543. And we made peptide remove one amino acid at a time at both end, N-terminal and C-terminal end, to see how each amino acid play a role for the clone activation. And you can find out different T cell clones can recognize independent amino acid. Some require L, some requires K, some requires C-terminal G. So slightly difference of a single amino acid change on a peptide region can select different T cell activation. And these clones can basically separate into main two groups, V-beta 5.2, V-beta 12 on the N-terminal, and the V-beta 4 at the C-terminal. And in disease, in diabetes NOD mice, there's expansion of V-beta 4 T cells. So the V-beta 4s for the C-terminals are more activated in the disease stage. So while in the N-terminal ones, they are expanded in resistant mice, NOR mice. So another example is this is the peptide. If you mutate one amino acid in this peptide at the K at the lysine 534 to tryptophan, you will convert that peptide, become agonist peptide for well-known T cell clone BTC 2.5. So clones with a single mutation can make a T cells without response to effector functions. Unfortunately, GAD65 doesn't have mutations in the islets, NOD islets. We checked that. So this tryptophan mutation is likely is a mimotope, is not a natural ligand that triggers this T cell response. So another thing I learned when I was in Eli's lab, T cells specific for the islet antigens, the repertoire is relatively small. By changing the repertoire is not sufficient to treat the disease. So when I have my lab, I explore new antigens. So how we explore the new antigens discovery? So I started from isolating microvesicles, or call it exosomes, from all kinds of cells really in the culture circulated. We first use the insulinoma called Min6 insulinoma cells. Those microvesicles are tiny, like a virus size. And if you use those vesicles, exosomes as an antigen to stimulate immune response in NOD mice, you learn that how the innate response and B cell response and T cell response react to those microvesicles. And surely innate is critical. Without innate MYD88 lockout NOD mice, they do not respond at all. So innate is critical. Without innate, there's no B cells, no T cell response. For B cell response, the peak reached at a 10-week-old NOD mice. When they go down in pre-diabetic or diabetic NOD mice. So the B cell responded early to the exosomes, and those B cells are mainly marginal zone-like B cells. T cells is typical T helper 1 T cell response to those exosomes because we measured interferon gamma, and the interferon gamma release as the interferon gamma T cells recognize the exosomes keep increase as far as the mice get in the diabetic stage. So at the 15-week NOD mice, interferon gamma T cells is the peak. So exosomes in those released by MIN6 cells contain unique antigens. They're capable to activate T effector cells that associated with autoimmune response. We also, this is insulinoma cells. We also isolate primary islet cells. If you culture islets, you can or axon prime tissues, you can get a mesenchymal-like stromal cells. Those cells release large amount of vesicles. And we also use those exosomes as a vesicle to stimulate the innate B cell and T cell response. Exosomes released by the mesenchymal stromal cells are very similar to the MIN6 exosomes. So the antigen, if there's any T cell antigen in the exosome, is not necessarily produced by the beta cells. The mesenchymal stromal cells can also produce it. So what are they though? So we performed mass spectrometry to detect the proteins in the exosomes. What is surprising is both the insulinoma cells and the mesenchymal stromal cells, they are enriched with endogenous retroviral proteins. And this mass spectrometry data, the list of peptides you can find in the exosomes, and this is the Western blotting of the MIN6 cells and their released exosomes. Those are the Gag protein, and these are mesenchymal-like cells. This is the cells, NOD-derived cells. This is the control cells derived from diabetes-resistant mice, stromal cells, Gag protein, envelope protein. This is the exosome. Similarly, only the NOD-derived mesenchymal cells express the retroviral proteins, not the diabetes-resistant B6 cells. So expressing endogenous retrovirus in the mesenchymal stromal cells somehow related to type 1 diabetes susceptibility. Among the different endogenous retroviral proteins, which one are good one to continue study? So Gag is the capsid protein. It's released as small viral particles. It can form a tightly dense particles like this one. If you see inside, they are densely packed Gag proteins. And those are the cell membrane, the transfected 293 cells. So they are viral size. They are like a virus, but they are not a virus, actually, just the Gag protein. And if you check Gag protein for HIV virus, Gag protein is the most conserved. They are like a rock. They don't mutate easily. They stay like conserved through many years evolution for endogenous retrovirus. So they are highly conserved. So, and we use Gag as a candidate to study T cell response by locking down the Gag from the insulinoma cells. If you use exosomes to lock down the Gag from MIN6 cells, and you produce exosomes do not have the Gag protein inside, and you use those exosomes as antigen to study T cell response, and clearly interferon gamma response is reduced without the Gag protein, and we also observe the Gag protein is required for early development of autoreactive T cells in NOD mice. When you transfer those T cells, they do not get, those ones recognize Gag is critical for type 1 diabetes development in competent in immunodeficient mice. So Gag is a complicated antigen. If you use the Gag sequence expressed in insulinoma cells to search the genome, you find many different highly homologous sequence to the Gag sequence. And they are across all different chromosomes. And some chromosomes has multiple copies of slightly different Gag sequences. They're highly homologous. This is all above 85 identical. And the majority of them are above 95 homology. So they're very similar Gag genes. in everywhere in the genome, and even some are reached in Y chromosomes. And then we clone what the GAG expressed in NOD islets, and trying to match the sequence to the genome sequence to see which one in the genome is associated with the GAG response in the islets. And unfortunately, after cloning all 16 different GAG clones, they are all different. They are slightly different though. They can be 98% homologous. So there's mutations in those GAG genes expressed in the islets. They are in, if you trying to match the sequence in the genome, it's a very difficult task to match where in the genome which GAG is produced, expressed in the islets. So how we cloning approach certainly is not a way to identify which GAG is associated with disease. And also there's many partial GAG could also produce proteins. So how we gonna find which GAG is associated with disease? So we developed a deep sequencing method. I should note that high throughput deep sequencing is not sensitive enough to identify small mutations. In the whole GAG gene has about 2 kb, and this high throughput cannot identify that, even with a few nucleotide change. So you cannot use high throughput. And we developed a targeted deep sequencing. So you amplify a short region in the GAG gene and perform deep sequencing. 1,000 sequences in that short region can be read. And then we analyze those deep sequences to find any motif or clues. So if you use serum or islets for RNA purification and cDNA, you do PCR amplification and you obtain those amplicons. You use those amplicons for either Sanger sequencing or deep sequencing. And some of them, Sanger sequencing, you can find those difference, but you definitely need a deep sequencing to read out individual mutations. Which region to target? This amplicon deep sequencing, so far the technology only limited to under 500 BP. So you cannot go, the gene is about 2 kb, so you cannot go that far. So you have to decide which region to target for deep sequencing. So we started from a known T-cell epitope region we mapped before. So idea is, okay, in that T-cell region, maybe some mutations associated with disease. So this region is at the C-terminus capsid region of GAG. And if you perform Sanger sequence, you can, NOD islets has those minor peaks. See the double peaks? You can see there's multiple genes in NOD islets, even in the same GAG amplicon. And in NOD, it's very clean. So the diversity in NOD is much higher for this region. So if you use deep sequencing, you can see how many different individual mutations numbers, and you quantify how many different variations, and which one coding for open reading frames in that amplicons. And by that way, you can analyze the mutation numbers, open reading frame numbers, and we see that NOD has higher variation numbers, and they have a lower open reading frames. So, and it can distinguish them NOD from resistant B6 other strains. So this is certainly is a risk prediction just by the sequencing of this amplicon. So then, can these markers, the variation and open reading frames, predict how fast the disease progress in NOD mice? So this is next question. We compare different age of NOD mice for the change of deep sequencing in that amplicons. And islets is difficult. Isolation islets is challenging. At least we see some trend in the islets open reading frame growing, but serum is much better clean data. So this is a variant number, 6 week, 11 week, 16 week. The number keep going up in NOD mice. And the open reading frame is the best marker to see there's more open reading frames keep going up in NOD mice close to type 1 diabetes. So open reading frame of Gag in this region is a marker. It can tell how fast the autoimmune progress to disease. So the right side is a longitudinal study. You keep getting blood from same mice from 6 week, 8 week, 10 week, 12 week, 14 week, 16 week. And you measure the variation numbers and open reading frame numbers in that same mice. The trend is so clear, especially the open reading frame can tell you it keep going up in these mice, in all the 6 mice we tested. So the Gag, the open reading frames is a good marker. Tell you the progress of the disease progress. So to study the mechanism, why these open reading frames is associated with disease progression. So the idea is, okay, this open reading frame may coding for mutations that can somehow activate autoreactive T cells. So how we test that? So we select those ones with open reading frames and translate them into amino acid. Then you align those open reading frames to see which amino acid is highly mutated. And in this amplicon at the capsid region, you can see out of the 120 amino acids, we find 14 highly mutated positions. Those 14 highly mutated positions contains all the variants sequence, because each sequence could have one mutation at the 316, another could have a mutation in 356. So each sequence variation could be different. How you can find which one in individual mice is associated with disease? So you have to do multiple mice at a different age. And we did that in both islets and the serum to see which amino acid mutations is associated with disease progression in that mice. Can we find public ones or common amino acid mutations that can be used as a biomarker or used as a target for T cells? So this is the hypothesis part of the mechanism study. So how can we test this? Endogenous retrovirus is involved of evolution of adaptive immune response, and the change we're getting the classic MHC molecules and the evolution from non-classical ones, and likely endogenous retrovirus may play a role for that evolution. So we hypothesize that non-classical MHC may be a good one to start with, because some peptide in endogenous retrovirus may be presented by classical one, non-classic MHC for T cell response. So we align all the genome Gag sequences, trying to find conserved ones. The idea is conserved ones may be presented by non-classic MHC molecules. And we do find multiple conserved peptide with a high affinity for one of the non-classic MHC called QA1. And these are 2 examples, and we call it the P2 region in the N terminal of Gag and the P9 region at the C terminal of Gag. And the P2 has very conserved in the genome, and the P9 has mutations in the genome. And we tested for these two regions of GAG. This is the P2 region of the GAG. So we use a IgQA1 dimer to stain T cells. And we can, using these tools, we can identify T cells, recognize QA1 presenting GAG peptide. It's kind of like a tetramer, just a dimer for presenting peptide to QA1 binding T cells. So in this way, we can detect GAG-specific T cells, and the P2 obviously is a good one. There's many T cells. There are quite a number of T cells recognizing the P2 peptide presented by QA1 MHC. And so we use deep sequencing to see the P2 region. Is there any mutation in the P2 region is associated with the P2 response, T cell response? Unfortunately, P2 is very conserved. P2 region really has rare mutations. So then we move to P9 region. P9 region is different. They are conserved in QAM binding from the T. So my mouse is, yeah. So the P9 region, the Q1 binding here, this is the Q1 region, they are conserved. Mutations mainly is the flanking residues, like the 316 and a little bit 325. Those are the Q1 binding region. So the flanking region is mutated. And if you do deep sequencing of the flanking of this region, and you find that the D316 is highly mutated. At 80, around 80% are similar, but the 20% is mutated. And this D is a good one if you see that different age of mice, 16-week, 11-week, 6-week, you see at the D region how far the mutation rate is for that single amino acid. These single amino acids can tell the trend. The mutant of the dominant mutant of the D316 is a glycine. So the mutation at glycine, you can track them. So single mutation, you can track them in the serum to see how the disease progression. And we did test the T cell response to synthetic peptide with D at a 316 or glycine at 316. So the glycine at 16 is more immunogenic. They induce more interferon gamma response for T cells. So this is a hypothesis-driven testing, is an ongoing study for how the mutations in the GAG affect T cell response. We are no new path in this way, but we're taking our effort trying to understand it. Thank you.
Thank you very much, Yang. Just wanted to see if anyone would like to have a question. Okay, I have a question. So if we think about genomic burdens and biomarkers, can you imagine that a composite ERV GAG score might predict disease progression in at-risk patients?
Yeah, we actually testing patient samples and compare with the healthy controls, particularly for human endogenous retrovirus K family drivers. And healthy controls and the patient are very different. So that's an easy task. You can distinguish healthy controls by reading out how many different open reading frames, how many different variants. So sure, the answer is yes. Healthy control is easy to distinguish from high risk.
Yeah, okay. And then, but could you actually have a score, like a graded score, do you think, or no? Not yet.
That will require a large number of patient samples to get uh, and testing multiple target amplicons.
Stages, yeah.
Yeah.
How many samples would you think you would need for that to really do it right?
Forget exactly score that as many as possible. If you just want a yes or no answer, that's easy. But if you want to really get a score to tell the risk, that's really needs a lot of patient numbers and testing multiple amplicons in the Gag
Mhm.
region, and maybe not just the K virus, uh, other
Yeah.
endogenous retrovirus too.
Others. Well, that leads me to my next question. Do you think beta cell stressors versus viral infection differentially drive these ERV rich exosome release?
For the younger ones, kids developing diabetes early at a very young age, I think that will be minor impact.
Mhm.
Maybe genetic has more impact. For older ones, yeah, I would think stress from islet beta cells it will play a role driving endogenous retrovirus expression.
Okay. Have you guys noticed that sort of exosome release inhibitors or uptake blockers in the NOD models dampened the autoimmunity?
Exosomes is a difficult one. Finding inhibitors. There are some related to exome release, but
Mhm.
it seems like we are studying a small subset of exosomes. We are focusing on the virus-like particles. They are
Yeah,
same size. They are membrane structures, so there's no way you can separate those virus-like particle from the exosomes
yeah,
because they are same size.
same size. Okay. Um, have you seen ERV signals in thyroid exosomes that shared etiology with the, you know, EAT and T1D?
That'll be wonderful to have have a parallel study for autoimmune thyroiditis, which is my PhD thesis. But uh, no, I didn't have a chance to study thyroid anymore.
Okay. Um, and what is sort of like, if we think about ERVs as sort of like a bridge, right, between viral infection and autoimmunity, what's the most urgent experiment sort of next year?
Well, testing T cell response is the ultimate goal, how that
Mm-hmm.
responded, and you never get satisfied by reviewers. Get a clean answer. Those antigens are T cell targets. So that's a that's a unfortunate uh, 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 fact. Uh, so I would think related to clinical mm, mm, is a better approach, urgent, which are the patients required. So biomarker is much easier than therapeutic. Uh, I would think uh, uh, as a new biomarker will be most urgent task to
Yeah.
study.
Well, biomarkers are definitely um, um, much needed in this space, especially sort of quantitative biomarkers or even biomarkers that could detect, sort of like waxing and waning, disease states. Here's a talk from Brian Liu. Thanks for the great talk. Could you share whether there are any endogenous antigens with or without mutation known to induce beta cell specific regulatory T cells?
We haven't get T cell, as I showed only one picture at the last slide, figure E. That's the T cell response. We're using mutated Gag peptide in NOD mice, and we haven't do any human T cell response to human endogenous retrovirus. So, as I said, T cell recognize endogenous retrovirus antigens is a known ultimate goal, but it's not an easy one.
Mm. Okay, great. This was such an interesting talk. Thank you very much, Yang. We really appreciate you coming on today and sharing these data with us. And we'll look forward to our next intersection with you, looking for your next paper. And thanks again. Have a great rest of your day.
Thank you. Thank you all.
Thank you. Bye-bye.
Bye-bye.